Yes. Presenter.
Good. Look forward to hearing your remarks.
You are?
Champ.
Ready?
Okay.
Good evening, everyone. How's the volume? We doing okay? Too loud? Good? Okay, just checking. We had some issues earlier. So I'm Angela Metler. I'm an administrative assistant in the president's office at South Dakota Mines. And welcome to Steam Cafe. We do this every third Tuesday. This is a partnership with South Dakota Mines, Hay Camp Brewing, and South Dakota Public Broadcasting. So we thank them for their support and thank you all for your support too. Whether you're a regular or a first timer, we appreciate having you here. I'd also like to welcome our online audience. This event is being live streamed. There will be a Q&A session at the very end of the presentation. So anybody in the audience, if you have a question, raise your hand. If you're online, you can ask your question in either the chat function or the Q&A function, and we will make sure that it gets answered live. Those of you who are regulars might remember tonight's presenter from last summer. She presented, I think it was in July, so this is kind of an update on her research. So Dr. Tanvi Govil.
Thank you to all. Can you hear me? So first, thank you to School of Mines and thank you to Hay Camp Brewery. Now it's okay. I love coming to these team cafes. If I'm sitting as an audience or if I'm presenting and having a light discussion over the drinks is something I really like. So as for my introduction, I have been associated with South Dakota Mines for almost 6 years now. I started as a PhD student. Then currently I'm working as a research scientist. And now from August 2022, I will be joining as an assistant professor. And before I actually jump into the topic of sharing my group's perspective on why we think genetically engineered microbes can change the industries of tomorrow, I'll give you, or maybe I'll have divided my presentation in terms of like a short story. Why we think that's important and what made us start working with genetic engineering. So when I started as a PhD student in School of Mines, my professors gave me a task. I have to design a process. That's first thing. It's like a manufacturing process. So I have to manufacture something. That process should be scalable. That means if I'm doing something in the laboratory level, later on, I should be able to translate it commercially. Let's say commercialize it. And like if you go to the right, there are big tanks of reactors. So start from a small scale level, move it to the upper scale levels, like a big tanks, big reactors, and subsequently commercialize it. Then they wanted me to make something which is close to a bioplastic, and that material should be made from a substrate or a source that we typically say that's procured locally. So first question, what bioplastic should I choose? So frankly, there is a lot of discussion and there are a lot of options in terms of what we say biopolymers or bioplastics that we can make. Example, they can be cellulose based, they can be polylactic acid based. What we choose to do is I choose that I will use microorganisms like the small bacteria. So bacteria in nature, they have an internal capacity to make a biopolymer or a bioplastic, which we call PHA. So assume like it's a system that camel uses. If you are throwing to the microbes a lot of carbon, but don't give them nitrogen, phosphorus, or stress them in certain conditions, so they start to use all the carbon they consume and keep storing them inside. Like if you see the microbes, so these are like tiny microbes that we work with, and those small granules inside are actually the PHAs that they have been storing. So they store these molecules under stress, but they also keep the capacity to use these granules, molecules, or we typically call them the storage energy modules. when they need it. For example, they stop getting extra carbon from the nature. So they start using their own food. So it's typically called a reserve food for the bacteria. That's first reason. And because the microbes make it, so assume in front of you like a Lego block structure. So they can be a red block, blue block, yellow block, so colors of different sizes and shapes. So these microbes have a capacity to choose the Lego blocks what they want. So the PHA molecule in itself, assume it can be one Lego block, it can be 10 Lego blocks, it can be even thousands of blocks long. And then there is a variation of what, like it can be, let's say, a red block repeated 10 times or 1,000 times. It can be a mixture of blocks. So this polymer is highly tunable. Basically, if you extract it successfully, if you commercialize it, then basically we can alter the properties of the plastics, whatever we need. Second reason we choose this polymer was it's 100% biodegradable in just a span of 80 days. Polylactic acid based polymers, cellulose is quite easy to be biodegraded. It degrades in just a span of 10 days. But lactic acid based, the basic biopolymer, if you go and buy industrially, is having a time span of 1 year. So we wanted something that can beat a time span of 1 year in case we need. And third thing is it has a huge market potential. So this bioplastic category has a CAGR rate of 16%. So that means that companies, industries do want us or as the researchers to make PHA-based bioplastics. So that's why we choose this polymer. Second reason, what substrate? So it's like giving food to the bacteria. I need to provide them some food to grow, to store these PHAs. And what we decided to use was a locally available substrate. We are using corn stover, the leftover of the cornfields. Why? Because it's a local source of South Dakota. Even in the Midwest region, if you see the map, there is almost almost million tons of corn stover available. Definitely farmers use it as a feed or a bedding for the cattle, but still after even using for their own supplies, they are left with much amount of stover still left. And if the farmers are selling them to us, it's an extra cost of income for them. Second, it being a non-edible part of the plant, so we are not scrutinizing any, the controversy over food and feed. Second reason why we choose corn stover as our substrate is, if you see this, I'll just quickly walk you through this table. So there are few companies, or probably I won't say many companies, who are making this PHA-based bioplastics. But if we quickly look at this third column, probably, so companies, basically they are using cane, beet sugars. So some sources, some substrates that are easy sources of sugars for them, maybe glucose. So some substrates that are very highly rich in sugars, they are using them. Other set of companies are basically relying on using oils that can supply a lot of fats to them. If you see, probably you won't see any company listed here worldwide that is using corn stover or any agri waste leftover as a feed material for the microbes. So the question would be why? And that's the thing that we thought for, that if we are successfully able to develop a technology, use corn stover, make PHA, maybe we would be able to compete commercially. Then, as I asked you, what's the reason that nobody is trying to use, or maybe they are using, but it has not been commercialized? So what's the reason corn stover has not been used commercially by any companies to make PHAs? So the reason is the structure. So assume like corn stalk or corn stover I'm talking about, it's a part of the plant material, and a plant has different layers. So if I again quickly walk you through the structure of the plant. So any tree, any plant material, first of all, what they have is an outer layer. Just assume there is an outer layer which is made up of lignin. And it's a very recalcitrant material, meaning it's very difficult to break that material. If there is anything that can break the lignin part or the outer shell of the plants. Then inside, there is availability for anything of what's called the easy source of carbon, which can be like, in our terminology, we call them hemicellulose and cellulose. So these soft inner cores are very rich in sugars. If you are using cellulose, they will provide you C6 or glucose, carbon atom that are 6 carbon long. If we are using hemi, they will provide you another set of sugars, which are basically, in our language, we call it C5 sugars. But before, let's say, my microbes or any microbes can have access to the soft cores, they need to break this outer core. And that's the main problem that has been faced by industries. So if I have to break the outer core first, I have to pass my biomass, or let's say the corn stalk, through a pre-treatment process. Means I have to treat my substrate with chemicals, maybe a high pressure, very high temperature. So a lot of chemical and physics goes into breaking the lignin part. Then once the lignin has been treated, it has been decomposed, broken out, then there is a second step. Second step would mean putting extra enzymes, something more extra catalyst, I would say, biocatalyst or catalyst, to now hydrolyze or break the inner core. So inner core, cellulose, hemicellulose, they still need some kind of treatment to allow you have access to the softer sugars. Like, so whenever we work in, the laboratories, our aim is to provide the microbes these final sugars. Once the sugars are, let's say there is an industry, so they'll first pass their corn stalk through the first step pre-treatment. Then they'll have a second reactor or a second setup where they'll do the hydrolysis or saccharification or breaking cellulose, hemicellulose, using probably a lot of enzymes. And then third steps goes into fermentation. Fermentation is the step now where microbes are going to eat the sugars and make the products they need. So no problem, we can always do that. You have access to physical treatments, chemical treatments, enzymes. We can just buy commercially and buy a whole gallon of enzymes. But multiple steps means multiple reactors, means investments. Second, it needs costs. Obviously, it involves cost. Third thing, it needs time. You have to first do first step, second step, third step, before you actually have the product in your hand. Then buying the enzymes is still an option, but it's costly. So I would say that there are a lot of companies that do use corn stalks, rice straws, a lot of residues coming from the agricultural fields, and they have been successful. But our thought was, can we design a process where we are eliminating 3 steps? Can we do everything in one single step? Find a microbe or find a catalyst, or maybe in simple language, find a solution that will do treatment for you. It will break out lignin. It will hydrolyze your cellulose inner core. It will make the sugars, and it will also make the product. So it's like a single step consolidated bioprocess. So that was the target or the aim. And if you go read more, that's the solution for a next gen technology. If you want to build new technologies that are cheaper, faster, or maybe actually using up the agri materials, we need to build up a technology that uses single cell CBP process. Now, maybe 2 questions was in front of us. Is it possible? Is it possible for us to design a CBP process that's a single step reactor? Is it possible? Second step, second question, if it is possible, what are the challenges associated? So now there is a category of microbes, bacteria that exist in nature, which we call extremophiles. Now those extremophiles mean they are weirdo types. They'll grow under high pressure, high temperature, low pressure, high extremes of pH, high extremes of anything that you can think for. So we relied on this for this project on a category of extremophiles, which we call thermophiles. So these microbes typically thrive at temperatures of more than 50 degrees Celsius. Why we relied on thermophiles? Because according to literatures and its proven technology, basically, thermophiles have lot of inbuilt enzymes like a catalyst stored inside them, using which they can actually break corn stover. They can actually break a lot of agri waste. They can actually break a lot of waste, if I would say, even a plastic waste. Then they, so in chemical reactions or chemical engineering, they also help us increase the mass flow rates, transfer rates. So basically, they just are very reliable sources to break any waste materials. So that's why we also thought to rely on thermophiles for our process. Now, frankly, question for us was, is there a thermophile that exists in nature that breaks const over? So yes, there are multiple options, or I would say there are few options available, where if you just feed your agri materials to these bacterium, they'll just degrade it. So that's a feasible technology. Even some companies are using it for ethanol productions. I don't know if it is commercialized, but in the literature, in the research areas, it's possible. Question for me was, does a bacteria exist that break the const over but also make the PHA? So one step process. It breaks the substrate, but it also makes the product. And the answer in front of me was no. There is no such microbe already existing in nature, which I can, just buy or ask the other researchers to give me, provide me. So what we had to do is we have to scavenge it. We have to try to find it by ourselves from the environment. So what we do now, so that's called bioprospecting, which we often call in our language. I want a microbe that can break waste for me. What could be the source they will survive? What could be the source I can find them? So I thought, let's go to South Dakota landfill compost site. So I went there, I collected some samples, and it's a very rigorous process of screening in the laboratories. We have to do some assays, some more screenings to find what microbes are living there, and then try to find out of, let's say, those thousands of microbes we found there, which is one bacterium, one microbe that we want. Now that 58 number is the ones that like unique ones. Unique means they were different from each other. But frankly, when I started finding those microbes, I almost started with 1,000 to 2,000 numbers. But at the end, almost like an year, we were able to find a microbe that makes the PHA, that also breaks the const over without me having to do any chemical treatment, without having us do any physical treatment, without having us add any external enzymes to it. So just take, let's say, a glass or a bottle in front of you, just take a small container, put your substrate. Substrate means your const over here. Put your bacteria, give them some nutrients to survive, and they'll do the job. And that's what we found. They were breaking the const over, they've been producing the PHA. So, and it's like a thermophile. It belongs to a Geobacillus genus, basically. And based on the project we were working on, so we named it Synambio 1 based on the project I was funded from. So one thing more, like it made, so first question probably, is it possible to design a process which is a single step consolidated bioprocess? Yes, it's possible, and we have the bug now. Second important thing that we found interesting, very interesting, was the material, the bioplastic this bacteria is making. It does not degrade till 420 degrees Celsius. It is so highly thermostable. The rest of the PHAs or bioplastics that you'll find in the environment, or sorry, the being made commercially, they basically degrade at a temperature close to 250 degrees Celsius. This one is almost having double thermostability. Maybe because it's coming from thermophile, we don't know. So those mechanisms are still under study. Why this microbe is making such a highly thermostable polymer. And because of the characteristics of this polymer, that's basically, we have patented this technology now. We have the bug eating const over, making PHA, and not only an ordinary or normal PHA, but something that's extraordinary thermostable. So as I said, it's the highest recorded thermostable and basically only the back. So we have not published the data yet, but when it's out, it's going to be the first microbes that going to have those advantages. So as I said, till now, everything is good. Everything is perfect. Everything looks like a dream world. Yes, everything is happening. So yes, yes, yes. My professor is happy with me. But am I, so everything is possible, but am I hitting the industrial standards? No. Why? Because the percentage of const over being broken down by synam biobacteria was just 26%. And the amount of PHA this bacteria was making or producing was just, assume it's in milligrams, 246 milligrams when I started. So if I'm using 5 grams of corn, I am making just a tiny amount of this polymer. So obviously next question for us was, can we improve this process? Can we improve the degradation percentage? Can we improve this? Yes. So like a typical, because I belong to a chemical and biological department. So yes, we have options, we have tools available with us. So I'll explore that now. That's the tools I'll explore in front of you one by one. So as an engineer, first step that comes in mind is let's start combining and permuting a process of finding the ideal temperature, ideal pressure, ideal nutrients amount, or probably then ideal home for the microbes where they are most active. So what we do is we try to change the temperatures, pressure, how much volume of my bacteria I'm putting in per liter of my reaction. So inoculum volumes, how much const over I'm feeding to them. So changing the rates of the const over we're feeding to them. And most importantly, how much time I'm continuing my process for, 5 days, 10 days, etc. So that's, in our terminology, we called it the process engineering design, process optimization. So just by finding the correct environment from the microbes, I was actually able to improve the production quantity of the PHA from 246, that was like kind of initial amount from me, to 342 milligrams. Still low, still in milligrams, but still an improvement of close to 40%. Then obviously it's not a satisfactory level. We have to further improve it. How to improve it? Probably the strategy one we have exhausted. This strategy might not help for us unless and until, I mean, this strategy always works. So there are multiple options. We can further keep on going, digging higher. But still we wanted to adopt a second strategy. So what we do, there is a concept which is called adaptive laboratory evolution. So I'll just try to explain it in simple language. So what you do is it's an evolutionary pressure. So we ask our microbes, so we like it's like an evolution, but under pressure. So we are trying to make them mutate. We are trying to make them modify themselves in nature by putting a kind of a pressure on them. How? For example, I want a microbe that starts to grow at high temperatures, let's say 100 degrees Celsius. So let's say my microbe just grows at 50 Celsius, but I want it to start growing at 100. So what I'll do, I'll keep on for generation and generation. I'll ask the bacteria, keep on growing at 100. So after generation and generation and generation, I'm forcing him to grow in an environment that was not natural for it. So that's called adaptation. We are causing the microbes to adapt in nature. Because we do it in laboratory, we called it laboratory evolution. So that's a simple technique, but a very powerful technique. And these days, there is a lot of investments on the industries and even researchers who are trying to rely on this technology as an alternative to strategy one that I shared with you, just basic permitting, combining the our parameters or conditions. What this technology is actually doing in my microbes, it is changing the genetic makeup of the bacteria without having me actually doing the manual treatments. So the adaptation, if it is working, it allows the microbes by themselves. It's a natural adaptation. So they will, maybe I'll explain this term simply. So there can be a single change in their genome. So just single change, single mutation, which we called. or they can even delete a chunk of their genome. So anything changed, modified in the genome that makes them genetically engineered. But I have not engineered them. It's a natural engineering that microbes have done for themselves just to adapt to the condition I wanted them to grow. Is there an advantage? So for sure. First thing, it is widely and easily applicable to any bacteria microbe you would be working with. You don't need a pre-information on what is the genome composition. Even if you don't know the name of your microbes you are working with, this methodology will still work for you. Does it need heavy instruments? No. Does it need heavy supply of enzymes, etc., or what we call anything like, does it need anything expensive? So no. You can just simply do it even in your like simple laboratories. But obviously there are challenges. What's the challenge number one? It's a tedious process. So what we have to do is we have to grow the microbes over and over generations. For example, complete the generation number one at 100 degree. So I'll start taking example in my study here. So I forced my Synam Bio 1 strain to keep on growing in presence of constover till I don't find a desirable outcome, or if I start to feel that, okay, now that's the limit that they can adapt, modify, and improve themselves. So I was constantly forcing them to keep on growing under, like with the presence of constover. I did it for almost 15 rounds, and for me, one round was of 10 days. So I grow them for 10 days, harvest them, again grow for 10 days, harvest them. So I was keep on continuing it for 15 days. Tough. It's taking time. After every round of generation, I have to screen them. Means, is there an improvement? Maybe, maybe not. So it takes time, it's tedious, and the chances of, you would ask me what are the percentage chances that if you adopt it, you are going to make it successful. So it's 50-50. You might gain the advantage, you might not. But in our case, I was lucky to find the advantage happening. First thing, I can further see an improvement in my PHA quantities. So I started, I would say, like, maybe don't go for this number, but close to 250 milligrams. And right now, after these two strategies, I was standing at 432 milligrams. Second thing, I'll come to those numbers later on. But the microbes, the enzymes, they need to break the constover. I can see by screening that those numbers were basically increasing for them. Sorry. So there was basically then improvement of the machinery. They were activating their machinery they typically used to break the constover. So they were getting activated. They were liking to grow in presence of constover. And it's a very basic thing. Means, for example, if I put in front of you a chocolate that we typically called, just give them very accessible source of sugar. We all would probably eat it in a 10 minutes thing. But if I put in front of you a food that you know it's inedible, kind of inedible, not 100% inedible, but kind of inedible, so you'll take time to eat it up. So our job was to activate the microbes to start liking that food. And that made a lot of difference to us. Then still we were unsatisfied. Still we wanted to do something else and something else, because ALE as a technology was taking time for me. It almost take me 1 year to finally screen. So in that 1 year, I also wanted to do something else. And that something else for me was now actually manipulating the genome, the DNA by myself. So having a targeted change in the genome, which I know if I do, I'm going to definitely see the improvement. I'm going to definitely see the change. Now, when I say actually what can we do, I can change the genome of the microbe. So what can I do? I can delete a portion of the genome. I can add some extra components to the genome, or maybe I can just modify or tweak a little their genome. How do we do that? Or probably where do we do that? So how will come later, but where do we do it? So here I would like to assume you, so even us, like as a humans, so all our information, how we function, how we work, how we eat, what our habits are, every information on how we function is stored in our body in chromosomes in the form of a DNA. So they can be, it's like a storage system. They are the databases of all the information that microbes, even as humans, we can have. That information stored within us can be stored in two forms. It can be a big database, which we call chromosome here. So it's a central database for the microbes where even like almost mega base pairs or mega amount of information is stored. Something at the raised to by like million level of information is stored. Then to help the big database, there are always some helping databases, which we in our language call plasmids. So they are just helping structures. And they have the capability to store kilobase, like something in kilobases, 10 raised to power 3 levels of information. What's the advantage of having a chromosome or having a plasmid? Chromosome is essential. If a microbe, if us, we don't have a chromosome, there is no information we can use. We are not going to survive. But is there an advantage of having a plasmid, like an extra database? Yes and no. Yes, it's just helping. So maybe if there are some functions that are not present in my own genome, let's say, I can always, add something or try to borrow from somewhere. Let's say I'm trying to borrow from you a gene, some more information from you. And if let's say I take something from you, added it to myself, it's easy for me to do those transfers through the plasmids because they are small. It's very difficult for me to do the transfers directly in the chromosome because it's huge. It's internally like present in a very stable form. And for you to, enter into the bacteria, microbes, have an access to those database of chromosome becomes tricky. So that's the advantage if your microbes have a plasmid, or you can add a plasmid, like an extra database to your microbes. But what's the disadvantage? And that's the thing that most of the time, if you have it's said that GMO microbes are not safe. They are not environmentally safe. So there is a controversy that don't use GMO. That's the problem. If you are doing anything at the level of just limited to the plasmids, they have a capability because they are small, and basically we call them transfer agents. They can come out of me, let's say, they can get transferred to any of you. You won't have any control on whom you are transmitting it to. Microbes do have certain limitations, or they have their own defense systems. I don't want to take you. I'm happy in my own way. But still, there are very high chances that these plasmids will force yourself inside you. But if I do any genetic manipulation in the chromosome, then it's quite possible that the changes will be only limited to me, or let's say my microbes. It won't be transferred to other microbes in nature. So as it is said, and you can beg to differ, but any genetic modifications done to the chromosome of the bacteria, they are 90% safe. Any modifications done to the plasmids, you'll have to put extra defense systems in place so that if you are releasing the genetically modified microbes in the nature, you are not harming the nature. So that's the basic thing that we were trying to do. So my target, or somewhere our target was, and I'll come to that part. So it's very easy for us to do changes in the plasmid, as I said, but it's very difficult to reach to the chromosomes, especially even with the thermophilic microbes. And I'll come to that point soon. Now, before I move to the next slide, one thing, if you notice, it is made up of DNA, even plasmids are made up of DNA. And if I try to explain for some of you what is a DNA. So DNA is a building block. So assume for now it's a script. It's a script present within us. Now they are made up of very simple building blocks called alphabets. So they are made up of script is made up of alphabets. So in the DNA terminology, we call them nucleotides. Those nucleotides can be A, T, G, or C. When those nucleotides starts to come together, they start to form words, which in our terminology, we call them genes. Then if you start to make phrases out of, let's say, 2 or 3 words together, then we always call them in our terminology operon, and so on and so on. If you just keep on adding words and words, you probably would be ending up a sentence. In our world, sentence means anything starting. Let's say there is gene number 1, gene 2, gene 3, gene 4. So there is a pathway that microbes have created in themselves to take X material to, let's say, a Y material. So they are transforming a starting material to an end product. And now if you can imagine, if I want to change, let's say I want to change this pathway. I want this whole pathway. I want it not to go from X to Y. I want this pathway to go from X to Z, let's say. I can change it at an alphabetical level. I can just go and maybe tweak A to G at some position. Just force the genome to have multiple G's, multiple T's, etc. I can go and change a whole genome, a whole gene. I can go and change the whole operon. I can even change the whole pathway. I can just delete the whole pathway. So what's the option for me? So if I'm asking, like I am going to manipulate the genome of my Synambio1 strain, what should I do? There are multiple options, and each microbe, I would say they are made up of 3.6 like into 10 raised to power 6, like millions of those alphabets. So which one should I change? So the answer lies in terms of, so that's called annotation. So we do a background study on the microbe. We try to understand the whole genome, what it is made up of, what's the pathway, or basically what's the portion of the genome they are making to make the PHA. So I am not concerned with what's happening in the rest of the world in the genome of a bacterium. I'm just concerned on the portion of the genome that makes the PHA for me. So when I was doing those annotations, I found that this bacteria has basically 3 pathways it can use to make the PHA. And those pathways are related to fatty, I would say the lipid productions. So when the microbe is making the lipids inside it, in some of those pathways, there are some loopholes and it goes to the PHA pathway. Now, if I delete the whole pathway, what is going to happen? I might end up having the bacteria no lipids, and lipids is like an extra layer for the microbes. It makes the outer cell wall, cell membrane for the microbes. So basically I'm jeopardizing the life of my microbes. What else should I do? So when we were doing the experiments, so we found that, if you remember, I told you initially, a microbe has a system to make the PHA, but at the same time, it has a system to even eat the PHA. They will, like by themselves, keep on eating it, consuming it. And we found that when it is a PHA-C gene, basically it's a synthase gene, it is making it. And if it is a PHA-Z gene, it's a depolymerase gene, means it's eating the whole PHA. So we thought that the easiest option for us for now will be, can we delete the PHA-Z gene? Just delete the gene that's making the bacteria eats its own PHA, because our target is not let the microbes keep on making those PHA, but also extract them at the later. So we want to stop the microbes from eating their own bioplastics, because we want to extract them and sell it commercially. So that was the thing, and a little bit some more results or studies on that. We actually found that when the microbes are making, like it's both the genes, both the enzymes, or what, let's say both the catalysts or both the tools are being used simultaneously by the microbes. It's not that it's first eating and storing and storing and storing and then start to eat it. No. So it is making the PHA, but simultaneously degrading it, eating, degrading, eating, degrading. And according to literature, it mentions that if we delete this gene, we can definitely improve the PHA yields, if not too much, by at least 20 to 40%. So we have the target now. But as I said, there is a problem. Problem lies in, because it's a geobacillus, it's thermophile. So in our terminology, we call these microbes non-model hosts. So I am not sure how many of you have heard, but E. coli is a very simple system. So there are microbes in nature which have different natures. Some easy manipulative, you can quickly go change their chromosome, and people have so much done already, so much research that they have all the toolkits available. You want to change the genome, go find it, buy it commercially, change it, done in a week. But this was a non-model microbe, means not many people work on them. Not many industries use them commercially, and definitely very few research groups in US, Europe, Asia that are actually trying to manipulate the genome of these tough microbes. So it's a non-model host for us. We do not have enough toolkits. We do not have enough knowledge on how to change the genome. So, frankly, I would say that, and I probably won't go into the details of, but there are two options for us to, if we want to delete a genome. One is a very popular technique called a CRISPR technology, and second technology, which we call as a homologous based recombination. So CRISPR is like a more advanced version of HR system. HR means I have to do each and every step of the gene deletion myself manually. Step 1, step 2, step 3, step 4. So there are steps associated. CRISPR is a modified technology. It is basically a one-step process. Same thing that probably we were trying to do. Can we convert a 3-step process to a 1-step process? So that's CRISPR here. But no tools were available for me in the lab to directly apply CRISPR to my genome or to my Synambio strain. So I, in my research, adopted a homologous recombination based system. So we actually developed the toolkit to do genetic modifications in this bacteria using an HR based system. And these are just the steps of, it's a multi-step process. And basically, if anybody of you is interested in learning this technology, you can definitely come to our labs. And if you also have so many of your students, or if any of your kids who want to learn with us these technologies, so my professor is right sitting in the back, so he loves inviting students to the labs. I think almost every summer we have 10 and 20 kids in the labs trying to learn from us something. So if you have somebody who wants to learn CRISPR or HR systems of gene deletions, we'll be most welcome to do so. So anyhow, after applying this technology, and this work is still in progress, but to date, I have already seen an improvement of 30% more in the PHA production yields that I am working on. So currently, if I would say my yields of the PHA stands at, so these were my previous numbers, and actually, I'm sorry, this should be 632, not 432, but my actually PHA number stands here at 632. We have achieved, let's say, the const over breakdown rate of 64%, and Department of Energy, NSF, any funding agencies, the average depolymerization rates reported in literature, reported by industries, without chemical, physical, any treatment, just stand at 25%. So our depolymerization rates right now 64%, and our PHA, I'm sorry, again, it's 632. That's now standing for us. And so what we are trying to do now, definitely there are improvements still going on in the background, how to further improve the yields, take this percentage not to 100, but at least more than 80%. Not maybe improve a lot, but at least take those numbers from, let's say, 632 to even like, if we can hit at least a gram. But we have also simultaneously started to scale up our technology. So right now we have a facility of running, like starting from small flask to 3-liter reactors, then 40-liter, 260-liter, and a 2000-liter reactor system is even going to come in, like it's in the so Poet company is going to set up a whole big, they're launching a bioproduct institute in the Brookings area and towards the Sioux Falls area. So the idea is to just keep on now scaling the technology. If we even scale up a technology at the current yields, we can make 1 kilogram of PHA in less than a week if everything goes perfect. Then, so that's the second part that we are starting to work now, and that's what I would be working as an assistant professor now. Second thing, no, that's the STEAM Cafe I presented in 2022. We have a high school student, a high school teacher, Donnie Decker. He came to our lab, spent almost an year with us, and we went to his farms. We found some more samples. We found some more microbes from his farms. And this is just an extension of maybe the knowledge I have gained through these 6 years, maybe the tools we have developed in these years, and maybe the new microbes we have found in nature. But we are even like now it's a separate topic, but we are entering into the bioremediation world. We are trying to break down pesticides. We are trying to break down PFAS materials. So PFAS, if you are aware or not, but it's the hardest materials in nature, the hardest chemical because of the carbon and fluorine bond. They are very prone to being persistent. They are not degradable. But we have found one microbe in nature that keeps the capacity. The genome is showing us evidences it can eat PFAS. And we might be engineering this microbe if we found that its wild type or parental type does not have, like, it's doing PFAS degradation, but it's still not at the level we want. So that's the second area of research that we are expanding, and we are using genetic engineering probably now. And then another, now that's a topic that's very new, and we have just got the funding to start this in School of Mines. So some of you still might have reservations on, you are still making a product using a bacterium, you are still making a product using a microbe, you're still making a product using a genetically modified organism, and they're not safe for the nature. Maybe your product is not safe for the nature because it's coming from a GMO bacteria. So we said, that's okay. We are going to take the whole genome machinery out from the inside of the body to outside in test tubes. That will be okay, right? We are not using anything genetically modified. We are just taking everything out from the body, whole chromosome, whole plasmids, everything this microbe needs, and produce everything in cell-free conditions. So that's the technology, or that's the process that we're going to now establish in School of Mines. Advantages, we have already found a microbe that can help us definitely, like even the same Synam Bio, it will help to make me PHA in the cell-free route. We have the options now to even make bio nylon, and we also have the option of now making PEEK. So PEEK peak is one of the, so somebody was telling me it's a polymer that has basically the highest strength in nature, and that's commercially produced only chemically. But we have the options now. We have the tools now, we have the knowledge now on how to make it biologically. Maybe it might need genetic engineering, but we are now trying to advance towards the route of cell-free system. Yep. So that's all I would like to share with you guys today. And I definitely acknowledge my PhD professors, my School of Mines, the funding agencies, SD Board, etc. And special thanks to organizers of Steam Cafe and Hey Cam Breweries. So any questions if you have, we can discuss. Thank you.
Go ahead and ask questions if you have any.
Go ahead.
You said that plastic, plastic breaks in the does it have to be exposed to something to break down, or doesn't break down in any case on the shelf?
No. So it needs a degradation atmosphere. So you can do it in soil. Just take your plastic, put it inside the soil. You can do it in composting conditions. There are options of degrading it in a marine condition. So, like a wastewater treatment facility. Definitely that 80-day number changes with the environment, but on an average, it's 80 days. So yeah. And that's what is another thing that we are trying to even establish in School of Mines now. So whenever, let's say, so frankly, we have not tested the biodegradability of our bioplastic because there is no commercial company available in South Dakota to do that. Even if we send the samples outside, it's costing us $2,200 almost per sample. And if I want to do it in replicates, it's just going to empty my pockets. So we are very much trying to have some fundings, or maybe applying for some funding where we can establish the biodegradation facility in School of Mines. So that time I would be able to give you more perspectives on what conditions we need to maintain to break the bioplastics. But in theory, it's degradable even in the soil. Go ahead. Yeah, so that's a good question, and that's why we are moving to the cell-free routes because of some Like I can, even if I try to convince you, I know I won't be able to take away that perceptions that genetically modified organisms can cause harm to the nature. These products can cause harm to the nature, but PHA is one bioplastic that is a certified safe. Even in our laboratories, I'm not doing that, my other teammates are doing. They're even trying to use it in biomedical applications. So they are making self-healing sutures or even the bandages using these PHAs. And during those testing, they have certified, or they are like initial results are mentioning that they are not harmful. So we are assuming that it won't be harmful or it wouldn't cause any problems to any kind of, like, it's environmentally safe. But as I said, it's like more discussions and more testings. But as of now, I would say it's pretty safe than other options. It's at least safer than using a plastic, means fossil fuel derived plastic. They don't biodegrade, they don't degrade, they persist, and it creates the problem of microplastics. So those microplastics can still exist with PHAs, if I can correct. Let's say if they don't degrade in the soil. Soil doesn't have the right atmosphere to break them. Microplastics still exist, can exist, but we have the options of controlling the microplastics. Thank you for the questions.
Bacteria you're using, so did you guys sequence it or no enough research and sequence, you know, already how to pick the
Yeah, so we sequenced it. We have a like a whole sequencing facility now in School of Mines. It's called a meta-omics lab, and my professor is the owner of that facility. So yes, we have the complete sequence of this microbe available, and we have even verified the sequence from 2-3 different, even we send it commercially, but we have also done it. So we have the whole genome availability. We have 10 minutes.
I have a question. At the very end, you were talking about taking the chromosomes out of the cell.
Not the chromosome. So chromosome will be always inside the, yes, in the later, yes, cell-free, yes.
Yeah.
Yes.
How does it survive when you take it out of the cell and put it in a tube? How does it function?
So that's a very intelligent question, because, every technology has its advantage and a disadvantage. So every time a technology comes up, it does has some, uncertainties associated. So cell-free technology, if you are talking to somebody, having a discussion, they'll tell you that thing, that it's probably unstable. If you are taking everything out from a microbe, they might be unstable. Your reactions, even if you are conducting in the test tubes, if I'm able to do it successfully in 10 days, like if I just grow the bacteria constalk for 10 days, even a month, they'll just keep on growing without inhibition. But if I take them out, there are definitely chances that we won't be in a viable conditions. But if you keep on supplying nutrients to even the chromosomes will survive. So if I ask you, so DNA is one of the, what's called, it has a very long shelf life, the DNAs especially. So if you know, like the fossils, the fossil that archaeologists go, so how do they find that this was the fossil of which animal? That's through the DNA. So DNA survive. They have the capability, even if you don't give them attention, they will persist. They won't lose their nature, except in Y conditions. So I would say that, to your question, that yes, there are chances, but that's science. So we'll have to make those conditions so that they are stable. And we are not concerned on the stability of the DNA. We are concerned on the stability of the enzymes, all the helpers that will make PHA, proteins, other polymers out of them. So yeah, very good question. So maybe if you are able to make a progress, your next team cafe next year would be on cell-free systems.
Anybody have any other questions? Anyone online have any questions? Nope. Okay. Well, in that case, like I said, we do this every third Tuesday. So next month, that date is, let me look. Don't remember off the top of my head. September 19th. And we will be hearing from the Crazy Horse Memorial Foundation. They're going to give us an update on the blasting and the fundraising and just kind of an overview on how things are going at Crazy Horse. So I hope to see you on September 19th. Thank you for coming.
Thanks.